In this video, we will work through an MCAT-style passage. To get the most out of this practice, pause the video and go through the passage and the questions on your own. Create a passage map by focusing on key concepts and relationships. This will help you stay focused and feel more confident as you work through the passage. These strategies will allow you to reach the correct answer more quickly and with greater confidence. With that in mind, let's begin the passage and good luck. Electron paramagnetic resonance, EPR. Spectroscopy is one of the few techniques capable of detecting radicals within living cells or inside enzyme active sites. In the past, the use of unsuitable standards led to many inaccurate results. Fortunately, the development of a new standard H at IBUT8 has proven to be a major success. The H at IBUT8 standard is produced by trapping a single hydrogen atom at the center of an octaisobutyl something nanocage. Figure 1. Here's figure 1. The structure of H at IBUT8. In EPR, radicals produce a sharp signal with a G factor of approximately 2. The G factor is a dimensionless numerical value that describes how strongly a particle's magnetic moment interacts with an external magnetic field. In EPR, the G factor is used to identify unpaired electrons and radicals in biological systems. The success of H at IBUT8 standard stems from its ability to remain inert across a wide range of temperatures. Its well characterized magnetic parameters over these temperature ranges, and the fact that its spectrum does not overlap with the spectra of other compounds of interest. Figure 2 shows the relationship between the G factor of H at IBUT8 and temperature. There's figure 2, and the caption says figure 2 H at IBUT8 G factor as a function of temperature measured at 9.48 gigahertz and 1 microwatts. Question number 1. A student is preparing a standard for EPR to analyze the radical responsible for type 2 diabetes. She successfully prepares the octabutyl nanocage. At the end of her preparation, she realizes that an extra hydrogen atom has escaped and entered the nanocage. How does this outcome affect the result of her experiment? Answer choice A says this outcome does not have any effect. She is able to carry out the experiment as expected. Answer choice B says the nanocage, now being heavier, has a reduced capacity to act as a standard. Answer choice C says the altered chemistry of the nanocage prevents it from behaving as a suitable standard. Answer choice D says a standard containing two hydrogen atoms at its center can act as a more efficient standard. Here's question number 2. In an experiment to create the EPR standard, a scientist observed that an electron in a hydrogen atom emitted a photon with a frequency of 5.7 times 10 to the 14 hertz. Which of the following graphs best represents this transition? Here's question number 3. In another study, it was found that a radical within an enzyme's active site exhibits the same G factor as H at IBUT8. What can be inferred about this finding? Answer choice A says this finding is implausible and the experiment should be repeated. Answer choice B says the radical is experiencing a similar electronic environment to that of the central atom in H at IBUT8. Answer choice C says the radical has an identical molecular structure to H at IBUT8 and is contained within a nanocage-like environment. Answer choice D says the radical chemically behaves like the central atom in the nanocage. Here is question number 4. In H at IBUT8, the hydrogen atom is trapped in a cage-like environment. Why does this improve H at IBUT8's usefulness as a standard for EPR? Answer choice A says hydrogen is a reactive atom, and the cage prevents fluctuations in pH experienced inside living cells or enzyme active sites. Answer choice B says it shields hydrogen atom magnetic signals used in EPR. Answer choice C says it lowers the free energy of the hydrogen atom, making it more stable. Answer choice D says it prevents hydrogen from interacting with other molecules. Here is question number 5. In the Bohr model of the hydrogen atom, which of the following electron transition leads to the smallest release of energy? And the answer choices are listed. Take a few minutes, go over the questions on your own, and then come back and see the solutions to the problem. On this slide, we're going to fully analyze the passage and create a passage map. The first paragraph does not provide many background science concepts that we have previously encountered. The main concept we may have encountered before is radicals, which are species with unpaired electrons, and this may be important to note. This paragraph introduces EPR and highlights the discovery of the new standard, which they refer to it as the H at IBUT8, which is described as a major success. So, strong phrases like this are important to pay attention to. The second paragraph shows the structure of the standard and gives more detail about what it is. We are already familiar with the hydrogen atom, which is mentioned here in the second paragraph, so we can note it and remind ourselves that we may encounter one or two questions about it. There's no need to fully analyze the detailed structure of the standard at this point. Just acknowledge that it exists and move forward. We can return to it later if it's needed. In the third paragraph, the passage introduces the concept of the G factor and it explains what it represents. This is new information. Here they say that the G factor is used to detect radicals and other species with unpaired electrons. In paragraph 4, the author explains why the standard is considered a major success and they introduce the figure. The main idea of the passage is that H at IBUT8, which was the standard they used, is an effective EPR standard and that it is an ideal for detecting radicals and unpaired species inside living cells. Let's move on to the questions and see how we can answer those questions together. So here's question number 1. It read, a student is preparing a standard for EPR to analyze the radical responsible for type 2 diabetes. She successfully prepares the octoisobutyl nanocage. At the end of her preparation, she realizes that an extra hydrogen atom has escaped and entered the nanocage. How does this outcome affect the result of her experiment? If you picked answer choice C, you are correct. Let's see why. You should know that when the MCAT presents a student's experimental work, in most cases, something can go wrong in one or more parts of the experiment. So always be on the lookout for that. Now, what is this question really asking us? It's asking what happens to the effectiveness of the standard if two hydrogen atoms are placed inside the nanocage instead of only one. The student was trying to prepare the standard for EPR. which is designed to work by trapping a single hydrogen atom inside the nanocage. That single hydrogen gives the standard very precise and predictable magnetic behavior. A single hydrogen atom acts as a paramagnetic species because it contains one unpaired electron. In this problem, an extra hydrogen atom accidentally enters the nanocage. This changes both the chemistry and the magnetic properties of the standards. When two hydrogen atoms are forced into close proximity, they can interact and form H2 molecule, which has paired electron and is no longer paramagnetic. And because of that change, the molecule can no longer behave as a proper EPR standard. That's the reason why the correct answer is answer choice C. The altered chemistry of the nanocage prevents it from behaving as a suitable standard. The key idea is that even very small changes in atomic structure can completely change how a molecule behaves in spectroscopy. If this question felt a little tricky to you, remember what we discussed earlier. First, figure out exactly what the question is asking, then connect it to your background science knowledge or the information presented in the passage. Keep that mindset, and you can handle any question the MCAT throws at you. Now, let's move on to question number 2 and see how we can solve that question. Question number 2 read: in an experiment to create the EPR standard, a scientist observed that an electron in the hydrogen atom emitted a photon with a frequency of 5.7 times 10 to the 14 hertz. Which of the following graphs best represents this transition? The correct answer to question number 2 is B, and let's see why. Let's solve this step by step. We're given the frequency of a photon as 5.7 times 10 to the 14 hertz, and we are asked to find which graph best represents this emission. We can round 5.7 times 10 to the 14 to the nearest whole number to give us 6 times 10 to the 14. We use the equation c is or speed of light is equal to frequency times lambda or wavelength. When we rearrange this equation, we get wavelength equals the speed of light divided by frequency. We plug in the values. The speed of light is 3 times 10 to the 18 meters per second, and the frequency is 5.7 times 10 to the 14 per second, or in unit hertz. When we round 5.7 to the nearest whole number, we get 6 times 10 to the 14. When we divide the speed of light, which is 3 times 10 to the 8, by the frequency of about 6 times 10 to the 14, we get a wavelength of about 0.5 times 10 to the negative 6 meters. And this value is about 5 times 10 to the minus 7 meters. 1 nanometer is about 1 times 10 to the minus 9 meters. When converting meters to nanometers, we get about 500 nanometers for wavelength. Now we compare the values to the graph options. The correct graph is the one with a peak at about 500 nanometers. The y-axis on the graph tells us the intensity, and the x-axis tells us wavelength in nanometers. So we need to pick the graph that has a peak around 500 nanometers, which matches answer choice B. Answer choice B shows a graph that peaks a little over 500 nanometers, which is completely fine because we rounded our value and we should expect to get an answer that is a little different from our calculations. So this is completely normal for the MCAT. That means the correct answer is option B. Remember, on the MCAT, it's completely okay to round your values to the nearest whole number. You're not expected to get perfect exact numbers, and that's completely fine. Approximate values are usually all you need. When you see a question like this, the first thing you should do is figure out what the question is really asking you. Sometimes it helps to quickly look at the answer choices because they can guide you toward where the answer is likely coming from. You can treat this type of question like a discrete MCAT question. Before doing any calculation, scan the answer options, decide on your next step, and do like some simple math. And then at the end, just match your result to the correct answer choice. Now, let's move on to question number 3. Question number 3 read in another study, it was found that a radical within an enzyme's active site exhibits the same G factor as H at IBU-T8. What can be inferred from this finding? Let's break this question down together. What is this question really asking us? It is asking us to determine what the G factor represents. We are told in the passage that the G factor describes how strongly a particle's magnetic moment interacts with a magnetic field, and that this value is used to identify unpaired electrons and radical species. We are told in the question stem that a radical inside an enzyme's active site shows the same G factor as the standard. The key thing to remember is that in EPR spectroscopy, the G factor reflects the electronic and magnetic environment of an unpaired electron. So, if two species have the same G factor, that tells us their unpaired electrons are experiencing very similar electronic environments, not that they have the same structure or chemistry. That's why the correct answer is answer choice B. The radical is experiencing the same type of electronic environment as the central atom in H at IBUTA or the standard. The other answer choices go too far by claiming identical structures or chemical behavior, which the G factor alone cannot prove. So the correct answer to question number 3 is answer choice B. Here's question number 4. It says in H at IBUT8, the hydrogen atom is trapped in a cage-like environment. Why does this improve the standard's usefulness as a standard for EPR? So again, what is this question really asking us? It's asking why the nanocage makes hydrogen more effective as a standard for EPR. The correct answer is D. It prevents hydrogen from interacting with other molecules. But why? Because in EPR, we need a signal that is stable and reproducible. By isolating the hydrogen atom, the cage keeps its magnetic properties consistent no matter what's happening in its surrounding environment. The passage specifically states that the standard success comes from its ability to remain inert across a wide range of temperatures, its well characterized magnetic parameters, and the fact that its spectrum does not overlap with other compounds. Since hydrogen atom is highly reactive on its own, since it only has one electron, the cage prevents it from interacting with other atoms, which ensures a clean EPR signal. Now let's look at why the other options are not correct. A says the cage prevents pH fluctuations. This is not mentioned in the passage, so it acts as a distraction. B says it shields hydrogen from magnetic signals. That would prevent detection, defeating the purpose of EPR. Answer choice C says it lowers the free energy of the hydrogen. The passage does not mention anything about the stability of hydrogen. So remember, the standard works because it keeps the hydrogen isolated, giving a consistent, reliable signal every time. The correct answer to number 4 is answer choice D. Let's move on to question number 5. Question number 5 says in the Bohr model of the hydrogen atom, which of the following electronic transitions leads to the smallest release of energy? Let's break this question down together using the hydrogen energy level diagram presented earlier. In the Bohr model, the energy levels are not evenly spaced. The energy gaps are larger when the levels are closer to the nucleus, and the spacing becomes smaller and smaller as the energy levels increase. Now, here's a key MCAT rule you must remember. The closer the energy levels are, the smaller the amount of energy released when an electron transitions between energy levels. Let's quickly check the answer choices. Transitions like n equals 2 to n equals 1 and n equals 3 to n equals 1 involve large drops in energy because these levels are far apart in terms of energy and are closer to the nucleus. The n equals 5 to n equals 2 transition still releases a moderate amount of energy. But now look at n equals infinity to n equals 9. At very high energy levels, the lines are very tightly packed together. This means the electron moves a very small energy distance between levels and it releases the least amount of energy. So the correct answer is answer choice D, n equals infinity to n equals 9 transition. On the MCAT, always remember transitions between higher energy levels release the least amount of energy. This concludes the video. For more review and practice, check out the other videos in this playlist or the links in the description below. Thank you for watching and best of luck to you.